Well, good evening, everybody. We're about to go live for the additional biology, followed by triple biology night before the exam hangout. Like we did previously, hopefully at this point, you should be able to hear me. And we're going to look at a few questions tonight, as we have done in the previous hangouts. So if you've got a calculator, a pen and some paper handy, it might be of use. And don't forget, if you're visiting us via Frog, you just post any questions you've got for me as we go through, and I'll try and answer as many specific questions as you've got. If I can just remind you as well, at this point, I'm not going to go through a huge amount of content. I'm going to show a little bit of exam technique, followed by some of the key concepts you're probably going to need for tomorrow. But if you have got a specific question that you're stuck on, if you just post it for me, and I'll try and specifically answer that as we go through. So we're going to make a start now. Before we get into specific points, hopefully your revision's gone very well. I can remind you at this point of the key topics that come up in additional science biology. They start with cells, where we need to know the structures of the cells. So for example, animal cell and plant cell and the parts within. We've got ourselves a little bit of cell specialization about how they're adapted for certain things. We've then got ourselves organization of cells, thinking about structures such as the lungs, the digestive system, the stomach with the villi, and we've got processes, so diffusion, osmosis, active transport. After that, we have ourselves organism distribution, so how to place a quadrat and a transect, how to sample for organisms, how to count them. You should also know a decent amount of plants, certainly what photosynthesis is, and we have seen in the past specific questions about labeling the structure of a plant and the vessels within. Fourthly, and probably quite a good proportion of the exam, will be about enzymes. So how they're used, how they help in specifically digestion, but also what respiration is and how enzymes are used within industry. And then finally, we have information on DNA, looking at the structure of it, the processes of mitosis, and if you're doing the higher paper, meiosis, how stem cells are used. And rounding everything off is a small but often found part in the exam, life on Earth. So looking at fossils or what causes animals to go extinct, and even new animals to evolve via speciation. So before we get into all of that, again, I'll be relatively brief this time, but please be aware for tomorrow, you've eaten lots of food, you've got a full belly, you're well hydrated, you've got some water. It's a very hot day today. If it's the same tomorrow, we want lots of water in. You're going to get a good night's sleep tonight. Try your best not to panic. Obviously, set the alarms, make sure you're where you're meant to be, make sure you know where you're meant to be with your seat number. And finally, make sure you've got all your equipment. Don't forget a calculator. It's a biology paper, but they often have a few questions in there with a little bit of tricky maths, so use the calculator. After that, some real basics, but again, pretty prudent. Know where you're sitting, know the start time. Don't forget you've got 60 marks in that paper. You've got an hour to do it, so it's around about a mark a minute. But you then need to be thinking about giving yourself some time to check your answers. Always look at the number of marks per question and answer it accordingly, if I can speak. Don't forget it's a real exam, so no silly comments. Don't leave any blanks or write I don't knows. Give an educated guess. Finally, from all of that, if you've got some knowledge up there in your brain about something specific. For example, a question comes up on photosynthesis. You look at question 2, it begins to talk about a plant photosynthesizing. Take a brain dump, write down everything photosynthesis needs. All right, start putting it down, find out what happens for it, and then look at the questions. Finally, don't forget, each question will have different components to it, and there's often some clues. So part C might well have some clues given to you during part A or part B. Okay, we're not going to look at all of these parts. Again, we can't be certain, but having a look at all the past previous papers, the additional science likes graphs. They put lots of graph questions in there. You're definitely going to get a 6-mark question. And they often give you some diagrammatic questions where you need to analyze things. It's not to say there won't be tables, but tables come up a lot more in core science than they do in additional. So I'm not really going to focus on those today, and I'm not really going to focus on circling answers, tick boxes, those kind of things, because hopefully by now you're well established. So let's get going. If you want to pause this as I go through, you're more than welcome to, and come back to it. We're going to take a look at some classic questions, and then I'm going to open up for the very end of this for a little bit of Q&A on specific things and my top tips. Once again though, before we go anywhere, don't forget, you've got your highlighter, use it. So this example of a graphing question, they've got some investigation. It's got bile on digestion, or fat by lipase. So we've got ourselves some enzymes, we've got ourselves the fat that's going to be broken down, a few other details. And they're going to mix some milk with bile in a beaker. They've got a pH sensor. They add lipase. They record the pH at 2-minute intervals. They repeat, and then they use water instead of bile. And we've got our results. We've got the pH here, and we've got our time in minutes. So if we look at this, we've got ourselves 2 parts to this graph. We've got with bile, where we're going from a pH that is dropping down and then leveling off. And then we've got one with water, which is following a very similar pattern, but it's just taking that little bit longer. Apologies for my drawing of the accuracy there. Okay, question 1, when we're looking at this, and again, these are questions to do with digestion, but will be coming up in the exam. This is why they've been put in there. And in fact, these are all from the 2015 or the 2014 papers, so they're all relevant, they're all relatively new. Question here, bile helps lipase to digest our fat. It's breaking it down. What is the evidence in figure 1 for this? Well, we're looking at the graph here. So we are saying, well, very clearly, that actually when we've got bile, it takes less time for the pH to drop. So we're getting our drop over less time. It's going quicker. And so we've got ourselves a faster reaction, or the digestion is happening quicker with the bile. Or conversely, it's taking more time with the water. You've actually got a mark for that there, but essentially you're saying that line is a little bit steeper. If we go on from that particular question, a second type of graph, a little bit more complicated this one, but the principle is exactly the same. And again, I'm going through it relatively quickly, but hopefully now you're well used to this. So we've got exercise, we're getting something to do with heartbeat, going faster and with a greater force. Just change my highlighter, it's not the best color. And then we've got a little bit of information. So we've got heart rate is the number of times the heart beats each minute. The volume of blood travels at the heart each time the heart beats is called the stroke volume. So that's the volume of blood traveling out. And they're doing an investigation to people running as fast as they can for a minute. They measure the heart rates of stroke volumes of person 1 and person 2 at rest during the exercise and after. And we've got our results. So if we have a look at this, and I'm going to take this off in a second, but we've got, first of all, our heart rate on this first graph. We've got this area is at rest. Then we've got this grayed out part during the exercise, and then after here is presumably post-exercise. If we have a look at that, we've got ourselves person 1, who is the dashed line, which I'm going to kind of draw in red, which goes up and down like so, roughly. And we've got this other person in blue that's doing that. It's going over slightly differently. I haven't drawn it particularly accurately. And like they've got for the heart rate, they've also got a second graph, a similar kind of thing, but this time it's for stroke volume. And again, we've got our two people. We've got person one doing this, and we've got person two doing this. So the question has a few bits of information before we get there. They're giving us cardiac output. It's volume of blood sent from the heart to the muscles each minute. And they give us a way to work this out. Something we've never actually done in the science paper, but it's given to us in here so we can just use this formula. So its cardiac output is our heart rate multiplied by our stroke volume. At the end of the exercise, person one's cardiac output is 160 by 77. If we have a look at that, if we look at person one for the initial cardiac output of a heart rate, we're around here, 160, and we've got the stroke volume, which is at the end of the exercise, just under here. And we're looking at just where the grey box actually ends. So it's relatively easy, this one. It's worth 3 marks, which is relatively generous in my opinion. But we need to then work out person two. Well, if we have a look at the graph for person two, their heart rate at the end of the exercise is here, which is about 148. Again, in the exam, this will obviously be nice and big for you to be able to see clearly. The stroke volume at the end is about here, which is about 38. And then all we simply do is multiply the two up. This is why we need our calculator unless you're very good at maths. And that equals 5624 centimeters cubed per minute. Now we're getting a very quick question then, asking us to use figure 7, which is why we're looking at this now as a graphing point. And we're asking to find out the main reason for the lower cardiac output of person 2. Well, the reason person 2 is lower is one of 2 options. Essentially, they've got a lower stroke volume. They've got less ability to push blood around. And so because it's lower, the number here, the stroke volume, is a smaller number. And so even if their heart rates are relatively similar, that big difference is going to cause the overall difference in the cardiac output. Okay, I've got one final question with that. And again, I've just copied the previous content across from over here. Person 1 is able to run much faster than person 2. Use information from figure 7 as well as our own knowledge. So what does the graph tell us? Well, the graph tells us that they've got an increased heart rate and they've got an increased stroke volume. So essentially they are able to send more blood around. They're pumping more often and they're pushing it out with a greater volume. That's going to get us 1, maybe 2 marks for that. But of course, now we're relating to what is actually in the blood. Well, the blood is carrying oxygen, it's also carrying glucose, and it's going to the muscles. But we're going to get 1 final mark for all of this, and that's the reason why our cells, our muscle cells, or in fact any cell in our body needs this blood with the oxygen and glucose within, and it's because they need to respire. And so if we've got more oxygen and we've got more glucose, we can have a greater rate of respiration. And respiration releases energy. So if we've got more energy, we're going to run quicker. So the marks from the graph come from telling us about the heart rate and the stroke volume. We're then relating that to our own knowledge, thinking, what has blood got? It's got oxygen and glucose. That's going to go to each part of the body, the cells that require it, so they can respire and release energy. Okay, very quickly, because it doesn't often come up, but don't forget, if we're looking at tables, use the information that's there. Again, I won't go through all of this, but essentially it's pH on digestion of an egg with an enzyme called pepsin, and they do a particular experiment. The question, at which pH does the pepsin work best? Well, we've got our pHs here, and we've got the length in millimeters of boiled egg white after 24 hours. So we're looking for the smallest length. The egg's been broken down. And we'll find that that occurs here, 20. Sorry, that wasn't the cleverest thing to do. There, 20. And that occurs at pH 2. Okay, 6-mark question. They do like a question on quadrats. It comes up relatively often. We've got to think about quadrats as well as transects. So let's take a look here on this particular part. For the 6-marker, don't forget we're going to get brackets of 1 or 2 marks, 3 or 4 marks, or 5 or 6 marks, depending on our spelling, punctuation, and grammar. You could do an absolutely perfect scientific answer, but if you don't have good spelling, punctuation, grammar, you won't be going into the middle or high marks. On this question, though, we're looking at ivy plants growing up trees and walls. Figure 2 shows us some ivy leaves. One leaf is from an ivy plant growing up a tree in the center of a shady woodland area. This is our center of woodland, shady woodland area. And we've got the other leaf from an ivy plant growing up a tree in a sunny area at the edge. A student makes the hypothesis the size of ivy leaves decreases as light intensity increases. The question, though, actually relates to how would we use the apparatus shown in figure 3, which is over here. We've got a tape measure, a ruler, and a light meter. How would we use this to test that hypothesis? And the important bit here, we need to make sure that we're going to get valid results. So we're going to get a few marks here. First of all, if we start saying that we're going to use our tape measure to produce a transect, and specifically, that transect is going to come out of the shady area and it's going to go into the light area. At that point, we then need to keep a couple of things the same. Now, these are growing up trees. So really what we want to do is we always want to measure at the same height of our leaves, same height up. So we're going to go up a given amount, whatever it happens to be, 50 centimeters, it doesn't matter, but we're always going to find leaves at that height. Again, this is going from a shady to light area, but there are other conditions to do with the weather. So we might think about the orientation. We might have it north, east, south, or west. We might always pick a leaf on one particular side. But when we've kept all that the same, essentially what we then do is measure the leaves, or for a more valid answer, multiple leaves, so 5 leaves at each place, perhaps, maybe 10. And we're going to use our light meter at the same time. So with the leaves, we will measure maybe their area to find out the size of them. And at the same time, we would measure the amount of light in a given amount for each particular place. And at that point, we could do a very simple job. We can have a graph with our information plotting the amount of light and the area of the leaf. And if the hypothesis was correct, we should find that as the light levels increased, we would have a decrease in ivy size, in ivy leaf size, something like that. So 6-marker. Use everything they give you in the paper. Check for all the clues. If they're giving you things like these diagrams here, it's for a reason. Check things that they're showing you, and of course, read certain parts. Okay, finally, and really quickly, if you're going to get tick boxes, these hopefully should be relatively easy marks, but again, look at the figures they're showing. So we've got a burglar. This is one of our DNA questions. They're breaking into something. They've got some broken glass. They've got some DNA taken from 3 suspects, A, B, and C. They're going to use DNA fingerprinting. Here's our broken glass. The question is, whose DNA does it most closely resemble? And hopefully, relatively early on, you'll understand that if you look at this, we are looking at B. For diagrams, again, one of the questions might be relating to photosynthesis with plants and the leaves. It might be the size of cells. In this example here, we've got ourselves a muscle cell, the wall of the stomach, seen through a light microscope, and they've given us on our actual exam paper a given distance, a scale, and they're saying that this figure is highly magnified. The scale bar in figure 6 represents 0.1 millimeters. Using a ruler, measure the length of the scale bar and calculate the magnification of figure 6. Well, this is relatively difficult for you to do on a screen, but on your paper, if you measured this, it would have actually been 4 centimeters in length. So 4 centimeters relate to 0.1 millimeters in real life. So all we then got to simply do is calculate that up. Well, 4 centimeters is 40 millimeters. And so if we've got ourselves 0.1 millimeters is relating to 40 millimeters, what multiplication gets 0.1 up to 40 millimeters? Well, if we times it by 10, it become 1 millimeter. Times it by 100, it become 40 millimeters. And so if we times it by 400, it will become 400 times magnification. And again, with diagrams, they like to give us lots of clues. An extinction question here, animal coming from fossils. We've got our Archaeopteryx here. It's living around about 150 million years ago, and there's a nice diagram of it. And if we've got a question, how is the fossil of Archaeopteryx formed? Well, again, there are some clues in this diagram. Clearly, the animal died. It's clearly been buried over time. And the harder parts, things like the bones, have not decayed. And what they've done is they have mineralized. So, animal dies, is buried, soft parts decay, but the harder parts don't. These harder parts mineralize, become rock-like features, and so we have a fossil. Now, our final question, classic genetic diagrams with Mendel. Here, they're publishing investigations about Mendel's pea plants, about the flower options, purple flowers versus white flower. Hopefully, though, you're remembering the generations. So we've got our parent generations, we've got our F1 and our F2. And if you remember, all of the first generation, when we had a particular cross, a homozygous, a purebred type, would become all dominant. In this case, they're all the purple color. The question here is, what is the ratio? Well, before we look anywhere else, hopefully you'll be remembering this F2 generation is 3:1, 75% to 25%. However, if you forgot that, again, the clue is in the diagram, 705 to 224. For every 705 of one, there's 224 of the other. So if you divide 705 by 224, you'll get a rough ratio of 3:1. You get the ratio 3.15 to 1. So roughly speaking, it's a 3:1 ratio. And then this leads on then to question 2 about why the large number improved it. Well, essentially, we're more likely to get our theoretical ratios. The more we look at, the more organisms we do, the more likely they are to work out to 3:1. In other words, anomalies are reduced. We might find that the first 100 are all white, and then the second 100 are all purple. And so the more we do it, the more likely we are to get these ratios, to get this valid information. And that's because fertilization, don't forget, is random. We get our alleles, we get a chance meeting. Okay, that's all the part now. Hopefully revision has gone well. I'm going to move on to one final part where I'm going to look at the classic questions that I think will be coming up tomorrow. If you've got any specific questions, please go on to Frog. Please just post them for me, and I'll do my very best to answer them as I go through. So let's have a very quick look at the key things that are likely to come up in the biology additional exam. Well, question one, the first set that I would make sure that I'm relatively happy with are cells. Remembering we've got our animal cells with a nucleus. They've got themselves mitochondria for respiration, releasing the energy. The nucleus containing the DNA. We've got our ribosomes, which make protein. Of course, they could ask cytoplasm, they could ask cell membrane. If we're talking about a plant cell, they've got everything an animal cell has, but we also have chloroplasts. Chloroplasts are for photosynthesis. Alongside that, we might have questions about different specific cells like yeast, which are microorganisms. They've got themselves a nucleus or cytoplasm, but they've also got a cell wall, or bacteria with no nucleus. Don't forget, we've got movement of materials in the lungs or in the stomach. We've got our cells diffusion. We go from particles in a high concentration to a lower concentration. It's the net movement. Now, they often go through semi-permeable membranes, so they allow certain things through and others not. Finally, if we're talking about cells, please be aware of specific cells we find in specific organisms. So if we're thinking about plants, we're thinking about palisade cells, lots of chloroplasts at the top, gives us lots of photosynthesis. We've got ourselves guard cells, which surround stomata, and they let in carbon dioxide, but in doing so, they also release water. We've got red blood cells that lose their nucleus at maturity. If we're grouping all the cells together, we would group them perhaps in the digestive system. So we've got particular cells like epithelial cells, which build up to make epithelial tissue. The tissues then all work together. They form up things such as the stomach, and all of the parts of the digestive system, all the organs work together, so the stomach, the pancreas, the intestines, to make the organ system. Outside of that, do not forget the photosynthesis equation. So plants need themselves carbon dioxide, they need themselves water, and they need themselves light. And that makes them some waste product oxygen, but also it makes glucose. And that is what is stored for energy. Outside of that, what affects the rate of photosynthesis? A whole host of things, but essentially it's the amount of light or it's the amount of carbon dioxide. If we increase both of those, they begin to increase my rate of photosynthesis, but it will begin to level off. This is because something else is limiting it. Now, going further from that, if we're talking about finding out how many animals there are in a given area, we might be using quadrats. So we'll be placing a 1 square meter quadrat down at random places. We've used a die to give us some random numbers, and we count the number of organisms found in those quadrats. We get an average. We then work out how many quadrats in total we could have fitted into the given field, and we also need to know the overall area of the field for that, and we divide them through. So if this was a whole area of 100 square meters, I've got a quadrat that's 1 square meter, so I could fit myself 100 of them in there. And if I found on average that I found 1 flower per quadrat, I would estimate there would be 100 flowers in that area. A transect, very similar. We use a tape measure for a line. It will go over different different places, so we might well have ourselves a river in between. But again, we place quadrats, or we count specific things at given areas, perhaps every 2 meters. We record what there was. So in this area, we'll probably find lots of fish. And funny enough, in these 2 areas, we'll probably find ourselves no fish, because there's no river. Or if we find the fish there, it will be a very unhappy, weird dead fish. Outside of that, we've then got ourselves enzymes. Like question on enzymes, they come up a lot. Biological catalysts, they speed up reactions, but they can denature because of pH or temperature. Essentially, they can break and not catalyze, not break things down. Be aware of the enzymes in our body. Amylase for starch to sugars, protease for proteins to amino acids, lipase for lipids to fatty acids and glycerol. Be aware that enzymes are really important in the digestive process. They speed it up. They're also important for respiration, and respiration is the opposite of photosynthesis. So we need our oxygen, we need our glucose, and when we have those 2, we convert it, and we have a byproduct of carbon dioxide, we have a little bit of water, and we also release some energy. Anaerobic, exactly the same, but we haven't got our oxygen, and so actually we don't make those 2, we make a little bit of lactic acid as well as our energy. Enzymes include biological detergents for industry. They change foods such as isomerases into glucose to fructose to sweeten them. Baby foods use protease to break them down. And then don't forget our DNA. We're going to be needing to talk about our chances of offspring, our genetic diagrams. So look at our Punnett squares. If we've got a male and a female, we draw our 2 parents, and then we simply cross them up. So we work down to begin, XX goes down, Y goes down, and then we go across, X goes across, X goes across, X goes across, X goes across, and we find ourselves with a proportion of 2 boys to 2 girls, 50-50. Be aware of the word dominant versus recessive. The dominant will show that characteristic in the phenotype, but the genotype could be both. So if we have an example here of something like cystic fibrosis, now cystic fibrosis is caused by recessive, so you could have a normal person who then has children with another normal person, but actually they were carriers of that recessive gene. They were absolutely normal in everyday life. They didn't have cystic fibrosis, but they were carriers of it. So if you do your Punnett square, we would have ourselves, and be careful of these terms, a homozygous non-carrier, 2 heterozygous carriers, but they're both fine, they don't have it. And unfortunately, like we have with Mendel in the question, a 25%, a 3-to-1 ratio, with one person having 2 recessive genes and suffering from it. With all of that, don't forget mitosis, cell division. We go from 1 cell to 2 identical cells. Meiosis, we go from 1 cell to 4 cells, each with half the DNA, which has been randomized, gives us slightly different offspring. And finally, be aware of fossils. The oldest fossils are at the bottom, and we have ourselves extinction, perhaps because of new disease, new predators, maybe some climatic change. And speciation can lead to the development of new organisms. Perhaps we have a population in one area, something happens to this particular population, there's some environmental impact that separates them, perhaps a mountain range, and we might find that this side remain as they are, that this side began to replicate and became slightly more triangular. Oh, this person's gone wrong. Yeah, he's an upside down person. And if they ever had their barrier taken away, they have changed suitably where they wouldn't then be able to successfully mate. It's a whistle-stop tour. I'm going to be online for about half an hour. If there's any specific questions, please let me know. I've just had one quick one about mitosis and meiosis, so I'm just going to go back to that in a little bit more detail for everybody. And we'll take that a little bit slower so we can go through. Mitosis will be there for everyone. Meiosis is likely to only come up in the higher paper. But let's take a start. So mitosis comes from a normal body cell. And this normal body cell will have a normal number of chromosomes. So we will have ourselves 23 pairs or 46 chromosomes. And in mitosis, the goal is to replicate exactly this cell twice. So what happens? Well, first of all, we have ourselves a cell with lots of DNA kind of randomly around. It's not dividing, it's in long strings. So the very, very first thing that happens is with this particular DNA, there's a signal that says, right, it's time to go via mitosis, it's time to duplicate yourself. And so the DNA begins to copy. And so each chromosome arm copies itself. So I'm going to draw a small blue and a long blue, and a small red and a long red piece of DNA. And what they will do, those chromosomes will duplicate up. They're identical. At this point, we then get ourselves a lining up of the chromosomes, and they line up perfectly. So I get myself my two blues, apologies for my drawing, but one is longer than the other, and my two reds. And these special spindles, these, we don't need to know this term, but these T spindles will come in and they will take 1/2 of each of my chromosome arms and then pull them apart. And as they do that, we're left with essentially, I've drawn that a little bit wrong, essentially a cell that's now increased its cytoplasm, and we have ourselves, it's not meant to look like a snowman, two parts like so. And what will happen very shortly is the membrane will simply close up, and we then have ourselves two identical copies of this first one. So we get a DNA duplication, we get a doubling up, and then we get a perfectly equal split. Now, meiosis is all about the gametes. And it essentially has the same start to the process. So we have ourselves a normal human body cell, but the gametes need 50% of the DNA. They need half of the information that we've got ourselves here in the original human body cell. So we've got ourselves our DNA. The very first part is we get a duplication of the DNA. And again, for ease, I'm going to draw just two. One's a bit longer than the other. So we've got ourselves a duplication. And like before, they line up and they're pulled apart. Now, what happens at this point, if I draw as I had before, my slightly extended cell, we're going to have ourselves, in this case, and again, I probably should have drawn that with them lined up actually before they're pulled apart, so they would have looked just like this. And what we find on this particular lining is that one side is pulled away and one side is pulled the other way. So we might get ourselves, for example, on this side, the two reds, and we might get ourselves on this side, the two blues. And then at that point, we get a second pulling apart. And on this case, we get a pull in a slightly different way. So we have, on this particular thing, the membrane falling out and the lining up. So on this one, I would have had my 2 blues, and on this side, my 2 reds. And again, because these are then pulled apart, we would find that we'd have ourselves from this particular one and this particular one, 2 more outcomes. So we might get ourselves our 2 blues, and we might get ourselves over here our 2 reds. But the crucial bit is they can be different, because we're getting ourselves here at the start a simple duplication, no randomization, but here we're getting ourselves a random movement. We're getting ourselves some random pulling apart, and this random part means we started with 100% DNA. We still have 100% DNA over here, but actually each one, because we've produced 2 from each part, only has 50% of it. And furthermore, it's random between the 50%. So we have ourselves a whole normal human cell that produces 4 gametes, each having 50% of the DNA. And the crucial bit is we get a randomization. Another question about the chromosomes: are they lines or X-shaped? They are drawn just as lines, so you'd have no problem just drawing them like so. But we often have in the examinations, we often draw them like this. It's not actually factually correct, because often they can look kind of weird. So we have lots of different shapes of them. But for simplicity's sake, we often draw them just as X's like so. But you wouldn't lose any marks, and I haven't ever seen a question where you'd actually need to draw the specific shape of the chromosome. Just going to ask if anyone's got any other questions, if you would like me to go through anything again. Now's the time to say, because I'm about to start the triple science. It's not a problem. I'm happy to stay on the line, and I can start them a little bit later. But if you've got a specific question for the additional part, please just post it on Frog now, and hopefully I'll do my very best to answer if I've made that clear. While I'm waiting for a few final questions, I guess my top tips for you all, if we're thinking about 6 markers, I think digestion is going to be pretty heavy in the additional paper. I think we're going to have lots of questions on enzymes. I think we're going to have lots of questions about how they're used in industry, the problems of collecting them, the expense, but the fact that they don't get used up. I think Mendel will come up with some genetic crosses. That comes up most years. I think you're very, very likely to have photosynthesis questions. Please make sure you're comfortable in drawing a plant cell, so you can draw the xylem and phloem, the tubes, the tape that you'll need for triple science, for triple scientists. But everyone else, they've got these 2 tubes. You've got guard cells surrounded by stomata, palisade cells at the top. And of course, the final part to all of that, just be aware of Mendel, animal versus plant cells, that kind of stuff. Oh, a very quick question: where is meiosis happening? So meiosis is happening in the gametes. So it's happening in the sex organs, the ovaries or the testes, and it's producing our gametes, our sperm and our egg. It still takes a normal human body cell But unlike mitosis, which occurs pretty much everywhere, apart from certain nerve cells, but pretty much everywhere, meiosis only occurs in the sex organs and obviously producing our half DNA randomized gametes. Okay, what I'm going to do, I'm going to stay on the line for the comment box. I'm going to begin the Triple Science Hangout in a second. So I'm actually going to stop the YouTube Hangout now. So I very much hope that was useful for you. Please have a good night's sleep. Please don't panic. I wish you the very best of luck. I'll see you just before you go into the exam. But I'll still be on Frog because I'm about to deliver the Triple Science part to everybody. So if there are any further questions, I'll answer them before I get going. Have a good night all, and I'll see you tomorrow. Bye-bye.